how Hodgkin and Huxley explained ionic mechanisms of action potentials. Before we knew how nerves actually worked, scientists knew nerves carried signals but didn't fully understand how. It was clear something electrical was happening, but the exact mechanism was unclear. Some thought it was just a simple wave moving down the neuron. That idea sounds reasonable at first, but it doesn't explain how signals stay consistent or how they restart at each point along the nerve. The real question became this: what exactly is moving during a nerve signal? Is it just electricity like a wire, or is something else happening inside the cell? This is where things started to shift, because the answer turned out to be more complex than a simple electrical flow. Why the giant squid axon was important. Hodgkin and Huxley needed a way to study nerve signals clearly. So they used the giant axon of a squid. It was large enough to insert electrodes and measure changes directly. That detail might seem small, but it made precise experiments possible. Instead of guessing, they could actually record what was happening. What they observed about voltage changes. When they stimulated the neuron, they saw a rapid change in voltage across the membrane. First, the inside became less negative, then briefly positive, and then returned to its original state. This pattern is called an action potential. The key question was what caused this sudden change? The role of ions instead of just electricity. They discovered that the signal was not just electricity moving like a current in a wire. It involved ions, specifically sodium and potassium, moving across the cell membrane. This is interesting because it means the signal depends on chemical movement as well as electrical change. Step 1: depolarization. When a neuron is at rest, the inside is negatively charged compared to the outside. During depolarization, sodium channels open and sodium ions rush into the cell. This makes the inside more positive. That rapid change is what starts the action potential. Step 2: repolarization. After that, potassium channels open and potassium ions move out of the cell. This brings the inside back toward a negative charge. The timing matters here. Sodium channels close while potassium channels open, which helps the signal move in one direction instead of going backward. Step 3: return to resting state. Once the ion movement slows down, the neuron returns to its resting state. Specialized pumps help restore the original balance of ions. This allows the neuron to be ready for the next signal. Without this reset, the system would not work repeatedly. Why this explanation was different? Hodgkin and Huxley did not just describe what happens. They created a mathematical model to explain it. They showed how changes in ion permeability could predict the shape and timing of the action potential. This was important because it turned a biological observation into something measurable and testable. What this changed in neuroscience. Their work explained how signals travel in a controlled and repeatable way. It showed that neurons are not just passive wires. They actively regulate ion movement to generate signals. This idea became the foundation for understanding neural communication, brain activity, and many neurological disorders. Final thoughts. What makes this discovery interesting is that it connects physics, chemistry, and biology all at once. A nerve signal is not just electrical or chemical, it is both. And once that became clear, it changed how scientists understood the brain at a fundamental level. It also makes you think about how something that feels so immediate, like a thought or movement, depends on tiny ions moving in and out of cells with precise timing.